morning, my dear students. Today we are going to be learning the process of recombinant DNA technology, that is shortly called as an rDNA. R stands for recombinant, and the recombinant is nothing but recombining. So recombining the DNA, that is called as a recombinant DNA. So this recombinant DNA consists of the different steps. So the first step is isolation of genetic material. So isolation of genetic material. The genetic material is DNA itself. Isolation is nothing but separating. So here one must know that the desired gene or gene of interest is present in a particular cell. That particular cell may be plant cell, animal cell, fungal cell or bacterial cell, any cell. From that cell, we want to take out only the gene of interest. So separating that gene from that complete cell by avoiding cell wall, cell membrane, cytoplasm, endoplasmic reticulum, Golgi complex, rest of all other cell organelle, we have to take only the DNA. That is called as isolation. So isolation is nothing but separation of DNA. So isolation of genetic material, it can be performed by the help of three sequential steps. It can be performed by three sequential steps. The first sequential step is lysozyme, is an enzyme used for bacterial cell. Imagine if the desired gene is present in the bacterial cell, we have to add a lysozyme enzyme to get that complete DNA. So isolating the DNA if it is present in the bacterial cell by adding lysozyme. Like the same, if the gene is present in the plant cell, we have to add cellulase. So by adding the cellulase, all the cell organelle and cell wall and cell membrane will become dissolved. We get only nucleus. That is the DNA. Like the same, chitinase is an enzyme which is going to be used for fungus. Lysozyme used for bacterial cell, cellulase used for plant cell, chitinase used for fungus cell. After adding this, we will get the DNA, and that is going to become suspended at the bottom by addition of chilled ethanol. This is a one mark question for your final examination. So after addition of a chilled ethanol to that medium, the DNA only get to become settled. Rest of other will get to become floating in that medium itself, because they are broken down, digested. This DNA is not only digested, it is a heavier weight compared to those, so automatically it will get to become suspended by addition of chilled ethanol. That is the first step of RNA. Whereas the second step is cutting of DNA at a specific location. When we get the DNA in a fragment form and that containing a gene, and that gene is going to be cutting at a particular sequence called as a palindromic sequence, and that is cutting by using the tool called as an REN, nothing but restriction endonuclease enzyme. So restriction endonuclease enzyme, after adding to this DNA, it is going to be cutting at a particular palindromic sequence. So we will get desired gene. That is the process here, right? Whereas the third one is that the desired gene is only one. We want to make it into the multiple copies. So making the multiple copies of DNA by the help of a process called as a PCR, that is called as amplification. So amplification of a gene of interest using PCR is the third step of rDNA technology. So amplification is nothing but making the multiple copies of DNA, making the multiple copies of the DNA by using a polymerase chain reaction. So that is going to be taking place by three sequential steps. So the first step is denaturation, second step is annealing, third step is extension. Once again, I am telling you that the third step is amplification of gene of interest using PCR. Amplification is nothing but making the multiple copies of the gene of interest using PCR is called as amplification. And PCR is nothing but its explanation is polymerase chain reaction. Right? So during this, the denaturation is the first process. Denaturation means nature of DNA is disturbing. What is the nature of DNA? Double-stranded coiled. Helically coiled and double-stranded is the nature of the DNA. And that should be become denatured. That is called as the denaturation. That is taking place by using heat process. When these double-stranded DNA will be given a heat approximately 70 to 80 or 90 degrees Celsius, the bond between two nitrogenous bases will become breaks. Then single-strand DNA will become formed. And each strand is acting as a coding strand or a template strand. They are going to be making helpful for binding another strand or preparation of another strand. That is an importance here. So, the denaturation by heat process. Then second one is annealing. Annealing means start making to extend the new strand of DNA on the old strand. This is the old strand which is going to be broken down into two separate strands by heat process. And this old strand is going to be developing into the new strand according to semi-conservative replication of DNA studied under molecular basis of inheritance, right now? So in that, the one strand is acting as a complementary strand and going to be developing a new one by the process of extension. And during this extension, two enzymes we are going to be using. One is a primer and another one is a DNA polymerase enzyme. And this primer is going to be binding here, and this DNA polymerase enzyme extracted from the bacteria called as a Thermus aquaticus. So Thermus aquaticus is the name of the bacteria available in the water which are surviving even at 80 degrees Celsius, because heat process is going to be giving at that temperature the enzyme has to work. So, their extension will take place and two strands of DNA is taking place by the process of amplification using PCR. So, another three more couple of steps were there. We will discuss it into the next video. Okay, thank you.